little preview of the lab that we're going to be doing tomorrow on photosynthesis. First of all, we are going to be using the chloroplast out of fresh spinach. And what I've done is taken the fresh spinach, put it into the blender with some sugar, 0.5% sucrose, ground it up, and spinach is very dark green, as you know, and so the chloroplasts are very easily extracted from it. The purpose of the experiment is to watch what happens in the light reactions. Okay, you know that in the light reactions, the final electron acceptor is NADPH. Well, in the we can't really measure NADPH, so in place of that, you're going to be using something called DPIP, and I'm not going to go through the whole long name because I can't say it anyway, but this chemical is blue. So you'll see it when you get it. It'll be in a dark bottle. It'll say DPIP, and you want to avoid light. Any light that gets on it can start to reduce it. When it's blue, it's oxidized. So then when it gets reduced with light, it becomes clear. So by using DPIP as our indicator, and that's all it is an indicator, we can tell how much light reactions are occurring. And then we're going to measure that using the colorimeter. Now the colorimeter measures color changes, and it does it at 635 nanometers of wavelength of light, and we're using red light. So you'll have a little cuvette. You'll put in there some phosphate buffer, some DPIP, and then you'll put in some chloroplast, and you'll measure how much light is actually absorbed. Okay, so remember, if it's not being absorbed, it's either being reflected or transmitted. So with this, we're looking for mostly we're going to be looking for absorption, how much of the light gets absorbed, and you'll get a reading after you put it into the colorimeter. Now, you're looking at two different variables on this. We're looking at boiled versus unboiled chloroplast, because we want to see if by destroying basically the membranes of the chloroplast by boiling them, what does that do for photosynthesis? Hopefully you're imagining that being boiled, that photosynthesis is not going to take place, especially the light reactions. Okay, then you're going to look at light versus dark conditions. So one of the little cuvettes that you're going to have is going to be covered in aluminum foil. So you'll cover the whole thing up with aluminum foil, and that will stop any light from getting into it. Once you get your three different tests set up, you're going to have to first do what we call a blank to calibrate. The calibration only needs to be done with before the initial colorimetry. You're doing it with light, you're doing it with chloroplast, but no DPIP, because the machine needs to be able to tell how much light gets absorbed when you have just chloroplasts in there, so that when the DPIP is in there and it's getting reduced, they can then calculate how much is then being absorbed. So you're going to be looking at boiled versus unboiled. You're going to be looking at light versus dark conditions, and you're going to be measuring that over time. So theoretically, the longer a little vial here, or I should say cuvette, with chloroplast and DPIP and light is there, and let's say the chloroplasts are not boiled, then you should see an increase in the amount of reduction. Correct? And you would expect that to go quite fast. If you boil them, then hopefully you should see no changes whatsoever. So we're going to be looking at light on boiled and unboiled. You're going to be looking at dark with unboiled, and so you'll be able to discern the difference. Now, just a few little things to remember, important points. First of all, do not allow the DPIP to be exposed to light. Be careful that when you add the DPIP that you're not sitting it in front of your little light that you're going to have there. And you're going to have a light, you're going to have a heat sink, which is going to absorb the heat from the lamp, and then you're going to have your cuvette sitting there. One of those is going to be in the dark, which you'll wrap it up in aluminum foil so that no light gets into it. You'll have a little piece of parafilm to put over the top. There are lids for these, but they're really hard to get off and on in a hurry, and they seem to be more of a hindrance than a help. Also, the chloroplast must be kept cold to prevent anything happening to the enzymes we want to keep, and we don't want them to be working too soon, basically. So you will keep your DPIP on ice, you'll keep your chloroplast on ice, and then when you're ready to begin, after you've done your initial calibration with the blank, then you're going to add your chloroplast and take the initial reading, times 0. At that point, you then put it under the light. And you're going to do this with each of your 3 different cuvettes that you're calculating with. Okay? Now, don't add any more than what it says to add as far as phosphate buffer, DPIP. Add exactly. These things are set so that they will work well. Watch your times. Get your times reading done as you're supposed to. So if you work carefully and you work and you watch your times, you should be able to get some good results. As far as analyzing the data, you're going to be able to print your graph. You're going to have to manually record your data and then add it into the computer, and it'll do the graph for you, and you'll be able to print it from there. So I think that's it, and it should be a fun lab, and we usually get great results. So see you tomorrow.